This is part 4 of the Chapter 8 lecture notes. Sorry, I hope this is getting everything in. All right, I'm just going to keep going, but it looks like it's cutting off some of the notes. But maybe it won't be too bad. Okay, let's look at number 9. What effect would a chemical that blocks the voltage-gated sodium ion channels in the excitable axon membrane of a neuron have on its ability to depolarize? What a question. Okay, it's just a lot of information, I know. Okay, first, let's review what depolarize means and the effect of sodium ions on depolarization. So depolarize means that the membrane potential, the resting membrane potential of -70 is going to move toward a more positive value. And it does that by allowing sodium into the cell. So from moving from the extracellular fluid into the cell, into the cytosol, sodium coming into the cell causes it to depolarize. So a chemical that blocks the voltage-gated sodium channels is going to block the entrance of sodium into the cell. So that's going to prevent it from depolarizing. It's going to have a negative effect on its ability to depolarize. Number 10, what effect would decreasing the concentration of extracellular potassium have on the membrane potential of a neuron? Okay, so we already have a high concentration of potassium inside the cell. If we decrease the concentration, then we're the concentration is going to be a lot. The concentration gradient is going to be a lot bigger. In other words, there will be more potassium inside the cell than outside the cell. There'll be a lot bigger discrepancy between the amount of potassium in the cell and outside the cell. And what that's going to do is cause potassium to leak out faster. And that's going to, potassium moving out of the cell causes the membrane potential to opposite of depolarize. It's going to cause it to become more negative. Okay? Number 11, list the steps involved in the generation and propagation of an action potential. And I won't go over that, but just remember what it takes. You've got to reach threshold. Graded potentials along the dendrites and cell body lead to the potential at the axon hillock reaching threshold. And once threshold is reached, there's rapid depolarization to +30. And then, and that's when sodium channels open, that causes that +30 millivolts. And then at +30, there is the closing of, or the inactivation of the sodium channels and the opening of the potassium channels. That's going to lead to repolarization back to resting membrane potential. Number 12, 2 axons are tested for propagation velocities or speeds. One carries action potentials at 50 meters per second and the other at 1 meter per second. Which is myelinated? Definitely the one that's faster. So that's going to be the 50 meters per second. Okay, we have actually already learned about a synapse. The synapse is where the neuron communicates with a target. In chapter 7, that target was a muscle, a skeletal muscle. And so we called the area where the neuron interacts with the skeletal muscle, we called that the neuromuscular junction. So this is some review here. But sometimes the target is a gland, sometimes it's a cardiac muscle or a smooth muscle, in which case it's still called a neuromuscular junction. But sometimes it's a gland and it would be called a neuroglandular junction. And then it can also be I think it's neuroneuronal. Let me see. It doesn't say. But you can also have a target that's a neuron. That we're not going to go into that because it's not in the notes. But the signals, the chemical messengers that are released across the synapse are called neurotransmitters. And the only one we learned about so far has been acetylcholine. But we do need to learn some terminology. So the axon terminal that contains those synaptic vesicles, that's going to release those synaptic vesicles across the synaptic cleft, that is called the presynaptic neuron. And then the target is called if it's another neuron, is called the postsynaptic neuron. Okay. Acetylcholine is one of our neurotransmitters, but here are some others. Norepinephrine or noradrenaline. Epinephrine and adrenaline are the same thing. Dopamine, GABA, and serotonin. Nitric oxide and carbon monoxide are gases that act as neurotransmitters. So there's more than just acetylcholine. Okay. Now, synapses in chapter 7, when we learned about the neuromuscular junction, that particular synapse was excitatory. Whenever acetylcholine is released, it causes an excitatory potential. And excitatory means that it leads to an action potential. But sometimes we want to inhibit an action potential. We don't always want action potentials just going off like crazy. We want to inhibit them sometimes. And so an excitatory potential will lead to depolarization, but an inhibitory synapse will lead to hyperpolarization, and that will inhibit an action potential. So some neurotransmitters and their receptors act together to lead to excitatory, potentials, and then sometimes they lead to inhibiting that potential. Okay, so usually acetylcholine and norepinephrine lead to action potentials. They're usually excitatory. Dopamine, GABA, and serotonin trigger hyperpolarization, which is inhibitory. But sometimes the same neurotransmitters that can be excitatory when they bind to one receptor can bind to a different receptor and be inhibitory. So it doesn't just depend on the neurotransmitter, it also depends on the receptor that it binds to. Okay? Let's look at, so the general structure of a synapse is that you have a presynaptic neuron that contains neurotransmitter, and then you have a target, which can be a muscle or a gland or another neuron, and the neurotransmitter is released across the synaptic cleft. It binds to receptors on the postsynaptic organ or target, okay? What effect, this is 14, what effect would blocking calcium channels at a cholinergic synapse have on synapse function? We didn't really go into that much. I can explain it this way. This is another way of saying, what effect would blocking calcium channels at a neuromuscular junction, like we saw in chapter 7, have on synapse function? So if we blocked calcium channels, remember it was calcium that led to a muscle contraction, led to an action potential, the neurotransmitter acetylcholine led to the release of calcium from the muscle, and that led to the muscle contraction. And so if we blocked calcium, there would be no muscle contraction. Okay, we're going to skip that. All right, you do need to know the meninges. The meninges are the 3 layers of specialized membranes that surround the brain and spinal cord. You may have heard of meningitis. My daughter's going to be a senior, and she just got a meningitis vaccine. And that's something that all seniors, at least in Wilson and Wayne counties, have to have in public school. So the 3 layers of specialized membranes are called the dura mater, the arachnoid, and the pia mater. And okay, these are small pictures. Let me see if I can blow them up. Here we go. All right, so this is the cerebral cortex, which is the brain. And what you're going to see is surrounding it and contacting it is the pia mater. And then we have the subarachnoid space. And then we have the arachnoid, which is this layer here. And then we have the subdural space. And then we have the inner layer, the dural sinus, and the outer layer of the dura mater. Okay? And then the spinal cord, we have the pia mater here, the arachnoid here, and the dura mater here. And then this is the epidural space, which contains fatty tissue, adipose tissue. That's where you get your epidural if you are having an epidural, if you're having a baby. So the dura mater is the tough outermost covering. The cerebrospinal fluid, you need to know this, cerebrospinal fluid is found in the subarachnoid space. And then the pia mater is the innermost layer. And this just says identify the 3 meninges surrounding the central nervous system: dura mater, arachnoid, and pia mater. And the pia mater is the layer that actually contacts the brain or spinal cord, and the dura mater is on the outside. Okay, I'm going to skip some stuff here because you don't have to know it all. All right, so let's talk about the brain. We already said the brain contains about 12 billion neurons. And then here are the 6 major regions of the brain. The cerebrum is the big part. That's all the little folds and stuff. That's the cerebrum, this whole big part here. And then there's the diencephalon, the midbrain, the pons, the medulla oblongata, and the cerebellum. Okay, so the cerebrum is this big portion. All right, and then here's the cerebellum right here, like a little knot in the occipital area. And then you have the brainstem, and we can see the medulla oblongata and the pons, but we can't see the diencephalon and the midbrain in this image. Okay, so the cerebrum is divided into cerebral hemispheres. The diencephalon, the left and the right hemisphere. The diencephalon is divided into the thalamus, the hypothalamus, and the epithalamus. The brainstem contains the midbrain, pons, and medulla oblongata. And the cerebellum functions in some motor activities. It has to do with balance and that kind of thing. Cerebrospinal fluid cushions the brain against physical trauma. It also helps in transporting nutrients, chemicals, and waste to and from the brain. And this shows you the flow of cerebrospinal fluid. I'm going to skip that part. So the cerebrum is divided into lobes, and they're named after the bones of the skull, so that's easy. The frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. And each lobe has sensory and motor regions. And you can see here, like the occipital lobe is the visual area. The parietal lobe is the somatosensory area. This is going to be the sensory area that relates with the skeletal muscles. So touching something hot and your skeletal muscles cause you to pull your hand back, different things like that. The frontal lobe is the somatic motor association area. primary motor cortex, gustatory